Hello, everybody. For our SURF 2020 research presentation, we will be doing it on interactive effects of benzophospyring, microplastics, and thermal stress on the species, a physiological model. My name is Adrian Castro.
Hi, my name is Marie Dandouche.
Hi, my name is Amber Henning.
And hi, my name is Ivana Smith.
And along with our mentor, Dr. Dilly, this is our presentation.
Organisms in the rocky intertidal zone live in a constantly fluctuating environment where they face a diverse range of challenges. For instance, there's wave action, increasing sea surface temperature, air exposure, competition, and predators, which are all stressors that these animals face simultaneously. Organisms in the intertidal zone, especially mussels, are exposed to air and a steep variation in temperature from the tidal cycle that induces thermal stress and leaves them vulnerable to desiccation, which means drying out. Mussels have adapted to be durable since they can be exposed to the air for an extended amount of time during immersion, specifically in the high and mid zone, making them equipped for the natural challenges they encounter on a daily basis. But now there are other stressors caused by human activity. For example, there is an increasing amount of microplastic and persistent organic pollutants in the marine environment. On top of that, the global sea surface temperature is also increasing at an alarming rate, which may be exacerbating the already stressful conditions that mussels and other intertidal organisms endure. Next slide. Why mussels? Mussels are key intertidal species that indicate the health and condition of the intertidal ecosystem, since this organism is so resilient. To translate, if mussels are not able to adapt quickly enough to the unfavorable conditions, then that means all other intertidal organisms are also not faring well. So mussels are filter feeders. So as you can see in the top bottom right picture, mussels will take in suspended particles in the water by passing large volumes of water through their gills, and then these particles will pass through to the digestive system. And these particles can be food like phytoplankton and algae, but they can also be microplastic particles as well. So these contaminants will accumulate within the gill and digestive gland tissue. And mussels being resilient filter feeders, sessile, and easy to collect make them amenable to lab conditions, and as a result, they are commonly used as a model organism for marine ecotoxicology studies. Since mussels are extensively studied, there are known biomarkers and analyses used to fully assess the stress responses and health of this organism by using the digestive gland and gill tissue. Some examples that we plan to implement in our study are using histology analyses where we can locate where in the tissue microplastics are and the associated cell damage. We can also assess the extent of oxidative stress by looking at the enzyme activity of catalase, superoxide dismutase, and glutathione peroxidase. We will even look at whole organism metrics to ensure that we have a holistic approach to fully study the health and condition of the mussels during our experimental runs. Next slide.
So to recap on what Amber was saying about the rocky intertidal zone, it's always constantly in flux. So mussels have to battle steep variations in temperature and long air exposure duration, all the while being sessile. And in addition, recent human activity has introduced new challenges for intertidal organisms, such as global climate change, which is warming the ocean, and exposing mussels to sublethal elevated temperatures, and inducing thermal and oxidative stress response in the organisms. Under sublethal thermal challenges, not only do proteins start to denature and unfold, but an upshift in metabolic rate is necessary for aquatic organisms to maintain homeostasis and protect against protein degradation. Consequently, increased metabolic activity gives rise to the formation of reactive oxygen species, which leads to oxidative stress. You might be wondering, what is reactive oxygen species? And it's a good question. They're oxygen molecules that are unstable and can cause cell and DNA damage. Think about what happens when you leave an apple out on the counter for a very long time. It starts to turn brown and it starts to get old really fast. Well, a similar thing happens to mussels when they're exposed to air for a long time. But don't be mistaken, mussels aren't helpless like apples on a counter. They're pretty resilient creatures. They have the ability to acclimate actively to their variable environment through their phenotypic and physiological plasticity, which can be mostly attributed to heat shock proteins. Heat shock proteins in the 70 kilodalton range, also called HSP70, function to maintain cellular homeostasis in muscles by folding and unfolding denatured proteins until they are properly conformed. So you can see why heat shock proteins are used in physiological studies. Not only do they help organisms during thermal and oxidative stress, but they provide further insight to scientists on the organism's energy depletion, metabolism, and thermal tolerance. And in addition to heat shock proteins as biomarkers for oxidative stress, famously, levels of antioxidants like catalase and superoxide dismutase stabilize reactive oxygen molecules, so they are also used to study oxidative damage.
Another one of the most prominent consequences of anthropogenic industrial expansion are the introduction of microplastics, which are plastic pieces less than 5 millimeters in size and can be seen on the right. Primary microplastics are introduced into the environment as tiny particles from sources such as microbeads in cosmetics or microfibers from clothing and textiles. Secondary microplastics are broken down particles from larger pieces of plastic, which can enter the environment through sources such as mismanaged plastic waste and abandoned fishing gear. These large plastic pieces are subjected to erosion from sources such as wave action, oxidation from sunlight, and biological degradation, causing them to break into smaller pieces. These microplastics pose a threat to many marine species, particularly filter-feeding invertebrates. Filter feeders do not selectively feed, and they're therefore unable to differentiate microplastics from food upon initial ingestion. Because plastic does not degrade after ingestion, studies have shown that microplastics have a negative impact on the food web, as they are able to trophically transfer through species and eventually even reach human consumption through seafood. Once ingested, they have been shown to have detrimental effects in marine invertebrates, reaching areas such as the gills, circulatory system, and the digestive gland, affecting organisms under both a cellular and transcriptional level, especially under chronic exposure.
Polycyclic aromatic hydrocarbons are a class of persistent organic pollutants and are one of the first airborne pollutants to be considered carcinogenic. In 1976, 16 PAHs were designated as part of the priority pollutants in the Clean Water Act. PAHs are the byproducts of incomplete combustion from anthropogenic and natural sources. Examples for anthropogenic sources can be from any industrial activity, incineration, or car combustion. Although car combustion make up bulk of PAHs in urban areas, natural sources can be from volcanism to forest fires. All PAHs are created equally, depending on their properties and the type of combustion processes. For example, BAP is created from low temperature combustion. Their fate in the atmosphere can vary also as well. So for example, they can either be deposited through wet or dry deposition, they can be transported through air or gusts, or they can be degraded or converted through chemical or photochemical reactions. Next slide, please. And so because of their aromatic structure, they're able to persist in the environment and transport through a variety of different environments and cause harm. So for example, they present a source of contamination to coastal environments from stormwater runoff. They are deposited into the ground and are washed away by water and go to the coastal environment. And they've been shown to have phototoxicity, genotoxicity, and oxidative stress responses to marine invertebrate when exposed to PAHs, and have been known to accumulate within the food chain as well. Next slide, please. So for our project, we're specifically looking at benzalpyrene. Benzalpyrene is one of the 16 PAHs designated by the EPA as a priority pollutant. They're also a group 1 carcinogen listed by the International Agency for Research on Cancer, which means that they are definitely known to cause cancer in humans. They've been shown to cause oxidative stress and genotoxicity in mussels. And another reason why we've chosen to include BAP as part of our experiment is because of their substantial presence in literature and also their well-documented effects on mussels. Next slide.
The increasing presence of BAP in the environment creates an additional problem when in combination with microplastics. Because of their hydrophobicity and high surface area to volume ratio, BAP is able to bind to microplastics, so they are able to act as a vector for environmental toxins to enter organisms through ingestion. The combination of BAP in addition to microplastics pose an increased threat to marine invertebrates as they're able to bioaccumulate and cause severe health problems. A few previous research experiments have examined either the dual effects of microplastics in combination with BAP, microplastics in combination with thermal stress, or thermal stress in combination with BAP. However, very few studies create a multi-stress scenario similar to that in the natural environment. And those that do have used different environmental contaminants and found that there is an increase in adverse response when 3 stressors are combined rather than any one in isolation. For example, while some studies say that microplastics may not pose that much of a threat to marine species alone, when combined with environmental toxins and thermal stress, contaminant concentrations have been shown to increase in addition to an increase in genotoxicity and mortality. To further study multi-stress responses, we plan to make an experiment using polyethylene microplastics, BAP, and thermal stress, which, to the best of our knowledge, has not been done before.
Okay, so just to summarize everything that we've talked about, essentially our goal is to study the integrative effects of POP-contaminated microplastics and chronic heat exposure in California mussels by quantifying thermal and oxidative gene expression response through qPCR, respiration rate, and a biomarker of enzyme levels. So we have come up with an experimental design as shown on this slide. And there are 3 experimental conditions. The first one is the control, which is just clean algae that will be fed to the mussels. The second one is microplastics that will be ingested by the mussels through a feeding mechanism using algae. And thirdly, we will feed the mussels with POP-contaminated microplastics along with algae. And so we will take these samples and we will subdivide them into 2 groups. The first one being 19 Celsius degrees. And the second one is a higher temperature, sublethal temperature of 23 Celsius degrees. We will then put those mussels and use them for our respiration rate protocol and then dissect their tissue, specifically the gill and digestive gland, which we will then use for analyses on gene expression and biomarker of enzyme levels. And so this will give us 3 experimental conditions times 2 temperatures times 2 types of tissues, which in total gives us 12 treatments.
So SURF has been an overall positive and helpful experience diving into what research will be like in the postgrad level. Dr. Dildy has been very helpful to us, has been showing us how to manage our time wisely, how deadlines will work at the postgrad level or at our jobs, and also how to manage our stress wisely when researching. During SURF, we were able to investigate microplastics as a pollutant in the environment, but also look at it as a vector for organic contaminants. We also looked at the effects of global climate change and what this will entail for all marine organisms in the future. We were able to incorporate all of these 3 stresses into a well-referenced introduction, while also creating protocols for experiment. And so after SURF, we plan to execute our protocols and methods, while also doing our post-experimental analysis, and eventually we plan to publish our work into a journal. I thank everybody for listening. I hope you guys have a great day.